in my MAP 2022 presentation and also in our new bioRxiv preprint, there are four main questions that we tried to address in terms of the sort of use of patient-derived xenograft models in preclinical oncology work. The first of those is how many patients it's possible to develop PDX models for. We know that there's a high degree of interpatient variability within non-small cell lung cancer, and we also know that it's not possible to develop preclinical models every time for every patient. And by taking this multi-region approach, which is really where our study differs from many of the studies that have been done before, we found that we were able to increase the proportion of patients within our cohort slightly, but still around 50% of patients in our cohort, we generated PDX models for around half of patients, meaning that for half of patients, we didn't derive a model. And that's still a major limitation, although it's a much larger proportion of patients than, for example, you would expect from cell lines. Interestingly, we're not entirely clear on what it is that means that patients either do or don't develop PDX models. So histologies are reasonably well represented between the two, between the group that do have a PDX and those that don't. Equally, there's no particular mutational subset of the disease that allows PDX derivation. And there are no obvious differences in the sort of patient clinical characteristics. We do find that tumor stage tends to correlate. So tumor stage and tumor purity, as judged by the whole exome sequencing, are both correlates of PDX formation. And we think that this means that there's got a long way to go still in our sampling of the lung cancer tumors, where we could potentially increase the number of patients that we can generate models for further still. The second kind of major aspect of the work that I presented was looking at how well the patient-derived xenograft tumor represents the primary tumor from which it was derived. So again, using this whole exome sequencing approach, we're able to directly compare the model with the region of origin. And by that, I mean a spatially adjacent region of the tumor that we've either sequenced or derived PDX for. And that study is very interesting because it shows that the PDX model tends to mimic the region of the tumor that it was derived from reasonably well. However, that it doesn't necessarily represent the whole tumor in the context of tumor heterogeneity. And so, in fact, you can have sort of very substantial genomic bottlenecks that lead to the derivation of a single clone from the spatial region being represented in the PDX model. And so the third thing that we were interested in is whether or not the PDX tumors continue to evolve during their time in the mouse. And so this is important because it means that even once you've established a model, we need to know that we can propagate that tumor in the lab, and that the results that we get at one point in time are still relevant at a different point in time, that the model doesn't continuously change. And our results regarding that are essentially that the tumors that do show some polyclonal engraftment tend to then become genomically monoclonal with respect to the primary tumor over time. But that the ongoing mutational processes in the tumor contribute relatively little to genomic distance from the primary tumor compared to that initial bottleneck of developing the model. And so ongoing evolution does occur in PDX models, but is not particularly, at least not during the first 6 months, isn't as significant event as the model establishment. And so our final question was to address the suggestion in the literature that maybe the patient-derived, the clones of the primary tumor that seed the PDX model might be representative of the clones of the tumor that go on to form a metastasis in patients. And we've been able to look at this in a relatively small cohort of our patients. But as you might expect, given what we're showing around clonal selection in multi-region xenografts, i.e. that different clones can engraft from the same tumour, it, perhaps unsurprisingly, is a mixed picture in terms of metastasis, in that sometimes the clone that engrafts does give rise to metastasis, but very often it is unrelated.